In this phase, some of those people who are not experienced in computational programming or any kind of coding, they normally face the problems, but I think it's quite easy if you could read a book about Unix command and awk commands. That will be very simple to write some small codes as a layman, and there is no need to understand some big or complex coding. In case if you want to skip this step, then you need to follow the or use the like software package, big package, that is for example here Galaxy, that is also called usegalaxy.org. So this can be considered as one of these software package for integration of multiomics data. Next step is here molecular docking and virtual screening. So this could be seen here. So for proceeding towards molecular docking and virtual screening, I have one dedicated lecture for molecular docking, which basically presented the idea of how to develop your own software or how to use the other software and understand the basic principle behind molecular docking. And then it is virtual screening. So this overall heading, molecular docking and virtual screening, is meant to simulate how small molecules could bind to target proteins, and it speeds up the lead compound identification. And the examples softwares are Autodoc, PyRx, Schrödinger Suite, SwissDoc, and CBDoc2 and some more softwares. This is kind of pipeline for understanding the drug screening approach. Now, the next part is sequence analysis and target identification. So it's quite pure bioinformatics and easy things for which you don't need to learn any kind of coding or programming. So here you could identify the genes or proteins involved in these pathways, and it helps in as a pinpoint novel drug targets. And some of the noted softwares are Blast, Cluster W, Omega, Ensemble, David, and T-Coffee, and some more. Then this is an sketch to present the single cell TCG as a spatial cell transcriptomics, single cell multiomics, then single cell data integration, free single cell sequencing, then single cell CRISPR. Then you could go for high throughput screening, target identification, pharmacokinetic evaluation, and finally you could go for Swiss admit or admit properties identification, and then apply its applications are for single cell technologies in drug discovery for tumor treatment or tumor eradication program. Then some of the like the specific softwares, for example, or databases are, this is PDB for protein 3D protein structure, and then UniProt is for primary sequence of any known protein. So these two are the most common softwares, databases, which are required for before proceeding for protein drug target identification.